Hey, Eric, it's all ready for you.
Okay, well, did you want me to say something about the slide or just start into with the,
No, just right there. People can read it. We can.
Very good. All right, so we're going to do a series of 10 talks. They're supposed to be short. I will be timing at 3 minutes. I will raise my hand on Zoom. And at 3:30, I will start trying to have you wrap it up. And then we want to allow a little time for questions. And hopefully that will make it so we get done with everyone within the hour. So I'm going to now share my screen. And put the first talk on. The first speaker is Sukiyama Amanu. And
I'm here.
great. And you can just say next slide. How's that sound?
Yes, sure.
Does everybody see it?
Okay, I can see it.
I think Gladys needs to stop sharing hers first.
I did. I can see.
Very good.
Okay. As long as we are ready, we will go ahead and you can just start when you're ready.
Okay, I'm gonna start now.
Okay, great.
Okay, good afternoon, everyone. Imagine going shopping for clothes and being handed a single one-size-fits-all item, regardless of your. Sorry, okay, 1 second. Sorry. Should I introduce or just like move on to the next slide? Because they told me that this slide will go away in a few seconds.
Yeah, as soon as you tell me to click.
Oh, sorry. Okay.
Yeah, sorry.
Okay, thank you. Next slide, please. Good afternoon, everyone. Imagine going shopping for clothes and being handed a single one-size-fits-all item, regardless of your size or style. It simply wouldn't work, right? Just as we all have unique sizes, cancer is no different, and one drug does not fit all. Some cancer resist treatment, much like trying to force like a wrong-sized outfit. What is the solution then? It's personalized medicine. Just as we tailor clothes to fit our unique bodies, researchers are tailoring cancer treatments to each patient's unique genetics. This overcomes drug resistance and improve the rate of success. It's like finding that perfect fitting outfit for every individual and represents the future of cancer treatment. Next slide, please. In the realm of precision medicine, machine learning plays a crucial role in predicting drug response based on gene expression data. Each patient's unique gene expression profile significantly influences how they react to different drugs, offering a real-time glimpse into which genes are actively engaged within their cells. Let's embark on a journey through this transformative process. It all starts with a training phase where our machine learning model carefully analyzes extensive data sets, including gene expression data from a diverse group of patients and their respective responses to different drugs. In our research, we delved into ovarian cancer data to illustrate this process. The model learns to decode subtle relationship between gene expression profile and drug responses, ultimately identifying which gene signatures are linked to positive outcomes and which are associated with drug resistance. Once the model is finely tuned through this training phase, it poised for practical application to new patients. By scrutinizing their unique gene expression data, it skillfully predicts how they might respond to a specific drug, thus offering valuable insight into the treatment most likely to be effective for that individual. Following rigorous validation, our model stands ready for real-world use. This newfound capability empowers oncologists to make more informed decisions about treatment options, significantly enhancing the chances of successful outcomes while mitigating the potential side effects. This approach marked a revolutionary in healthcare. In fact, we achieved an outstanding accuracy rate of over 90% in identifying the genes responsible for resistance to platinum chemotherapy in patients with serous ovarian cancer. It's a departure from the traditional one-size-fits-all approach, propelling us towards treatment tailored precisely to each patient's unique biology. This holds the potential for more efficient and precisely tailored therapies that not only minimize side effects, but also optimize the potential for successful outcomes. In our journey, let us always remember that in healthcare, one size does not fit all, but instead personalized medicine stands as the guiding star. Thank you.
Right. Well, thank you very much. Does anyone have any initial questions? Questions for this about this talk? Well, I mean, I definitely have one.
Sure.
So I'm just curious about, I mean, you hear about this stuff in the news about tailoring drugs to different types of, especially cancer, you hear about this, but like, do you imagine, knowing a little more about this or much more about this than I do, do you imagine that this would be something that's sustainable, that you imagine like in 20, 30 years, like a number of medications will be developed for individuals based on their genomics? Or is this something that's going to remain like kind of a niche thing for very hard disease?
I think it can work because the issues that we're facing these days is just the availability of the data, because not for machine learning model to work, it needs more data to validate the results that we get. And if researchers get more and more data, then it'll be really helpful to validate the results that we get, different researchers get, and that definitely will help the oncologists, because so far what's happening in the healthcare is that we have a panel that has very small amount of genes that contribute to the progression of those ovarian cancer, and so testing those small amount of genes are not enough to decide whether that drug will work or not for that particular patient, because we have different genetic profile, and we have, one drug. That's why I mentioned one drug does not fit all, because giving one type of drug or one type of chemotherapy for different type of patient who has different type of cancer, each cancer has different subtypes and different stages, different grades. So by optimizing, by doing more research and finding these biomarkers that really affect in the the efficacy of the chemotherapy, and trying to find those drugs that really play an important role in the pathways that cancer affects, it will really make a change, of course, in the 20 or 30 years. But by just focusing on those 12 genes in that panel, definitely it will not help us to find a solution for cancer.
Got it. Thank you. Any other questions? Time for one quick one. I think Gladys has her hand raised.
I was just applauding silently, Eric.
Oh, it's applauding. Sorry. Okay. I'll get the hang of this. Okay, so thank you very much, Sukiana, sorry.
Thank you.
I'm going to go pull up the next talk. And Grace wanted me to mention that the rest of the talks are now up for voting for our award for best presentation, I believe. It's the rest of the talks, so please be thinking about that when you view the rest of the talks. Okay. Here's the next one. This is from Eleni Adam from Old Dominion University. Go ahead when you're ready.
Hi, everyone. My name is Eleni. I'm a PhD candidate in the Department of Computer Science at Old Dominion University. And today I will present to you our research titled Analysis of Subtelomere Telomere Regions of Metastatic Cancer Genomes. So our work concentrates on the human genome. Each human chromosome has 4 main regions. The telomeres at the tips of the chromosomes. Here in the image, they are designated with white color. They're also known as the protective cups of the chromosomes. Then we have the subtelomere regions located next to the telomeres, the non-telomeric regions, and the centromere. The telomeres, despite the fact that their length is less than 1/10,000 of the total DNA in our cells, they are a vitally important part of the chromosome. Their dysfunction caused by length shortening can lead to senescent cells, which in turn cause inflammation, resulting in age-related diseases, including cancer. The subtelomeres possess the critical role of regulating the adjacent telomere lengths, and for that reason, we focus on the telomeric and subtelomeric regions of the chromosome. In order to study the genome maintenance in cancer, we use part of the TCGA, the Cancer Genome Atlas datasets, which has 33 cancer types. And specifically, we started with the prostate cancer data. These datasets are all very large. Each genome is about 1 tera in size. And therefore, when you have hundreds and thousands of these to analyze, the size grows really fast. And so do the resources needed for the analysis. it would be impossible to do it without the use of high-performance computing clusters as well as the clouds. And for that reason, we use the Old Dominion University WAHB and Turing clusters and the Amazon Web Services Health Omics infrastructure to analyze the huge data sets. Next slide, please. Okay, so our pipeline consists of three main parts. In the figure, we can see the first part, which is the use of computational methods to extract out of an entire genome dataset the parts that correspond to the telomere and subtelomere regions that are of interest to us. So as an example, for one genome, we start with about 1 tera data, and after about 27 hours and use of multiple software tools, we obtain the data that are related to the subtelomere and telomere regions of one genome. Imagine that the metastatic prostate cancer dataset that we have started with has 101 genomes to analyze. In the second part of our analysis, we define the subtelomeric features, which then in the next step, we apply machine learning methods to correlate them with the clinical outcome as well as past research discoveries. The pipeline is implemented using Nextflow, making it available for direct use under any environment. Our goal and purpose is to understand cancer mechanism better in order to improve our ability to diagnose cancers at an earlier stage and treat them with more specific and effective therapies before they reach the metastatic cancer stage. Thank you.
Thank you. Well, do we have time for a question? Well, I would just start. I would just like to, just my own selfish reasons, I'd like to know roughly how much you did on the WAHB cluster versus AWS. Roughly.
Oh, yeah. In the WAHB cluster, we started experimenting with one genome, and we essentially built our pipeline there, did some tests, and but of course, when you're using a cluster at a university, you're sharing resources with others. And even if you do allocate parts of the node, you still have the memory, which might be used by another person. So the software might take longer. So we did notice a difference in time when we moved to AWS. And of course, you have all their sources available over there to run 100 genomes at once. So that was the main difference, I guess.
Are there any other questions before we move on? Okay, well, thank you very much.
Thank you.
Okay, we'll have the next the next presenter is Rebecca Fogarty from Virginia Tech. Let me pull that up here. Okay. Please go ahead. You're ready.
All right. Hi, everyone. I'm Rebecca Fogarty. I'm a second-year master's student in the Lemko Lab at Virginia Tech, and my project focuses on computational investigations of oncogene nucleic acid structures. Specifically, today, I'll be talking about the PIM1 serine threonine kinase. So PIM1 is overexpressed in a wide variety of cancers, including many reproductive cancers, as well as the highly aggressive triple-negative breast cancer that is super difficult to treat due to very limited viable chemotherapeutic targets. So we can see here that PIM1 overexpression leads to a variety of sort of downstream cellular events, all of which contribute to tumor growth, progression, and metastasis. So the idea here is that potentially through stopping this PIM1 overexpression and overactivity of this kinase, we can then therefore target this tumor progression directly. So the PIM1 oncogene has been found to have two distinct G quadruplex structures form in its promoter region, shown sort of on the bottom center of the screen here. And promoter G quadruplexes have been found to play a key regulatory role in gene expression. So the basis for my project here is that potentially through stabilizing these G quadruplexes, we can inhibit that PIM1 transcription and therefore develop a new chemotherapeutic strategy for patients that are lacking effective strategies. So I explore PIM1 through use of molecular dynamics and enhanced sampling simulations. And both of these methods sort of provide us insight on the conformational, ensemble, and free energy profiles of these structures. As you can see sort of in this structure here, large conformational change, as well as electronic properties. My group really likes to focus on ion activities within the structures, as well as any electric field analysis that we can do, as both of those are really vital to take into consideration for further drug design. However, many molecular dynamics methods actually lack the ability to appropriately quantify those values. Additionally, the latter half of my project focuses on in silico screening methods. As you can see over on the far right of my screen, we will develop, or through use of the method SILCS, which is a fully computational screening method, we can actually develop these frag map sort of visualizations that provide us insight on sort of which regions of our structure will actually bind certain characteristics of common ligands. So you can see it can identify regions that apolar sites bind to, as well as hydrogen bond donors and acceptors. And the insight that we gain through that will potentially allow us to develop knowledge on how we could potentially target form 1 over form 2 of this G quadruplex, as these PIM1 G quadruplexes are really relatively new and are currently being ongoingly studied in regard to their biological role of both of the forms. So with my data, as well as, again, future further studies, we can potentially develop a new and more specific chemotherapeutic strategy for these patients that typical methods don't work for. Thank you.
Well, thank you. Sorry if my lowering hand isn't always working. That seemed to work there. Are there any questions on this? Of course, I have one, but please, someone else first.
I'll go ahead and ask. Thank you, Rebecca, for your presentation. Regarding the middle box of PIM1 overexpression. So, if I understood correctly, PIM1 is the oncogene, and it causes, among other things, inactivation of a apotopic protein. I'm trying to remember how to pronounce apotopic biology. And I'm guessing that this causes cancer, and that like apoptosis is the procedure by which cells for lack of a better term, kill themselves, or like die off like naturally. And so inactivating this prevents them from dying off, which then contributes to growth of like masses that can become cancerous and take nutrients from the body. Is that correct?
Yeah, exactly. So that inactivation can lead to basically immortal cancer cells, so that they can continuously proliferate throughout the entire progression of the disease.
Okay. Fascinating. Thank you.
Rebecca, what sorts of codes and tools did you use in this analysis? I was just curious.
Oh, okay. So we use primarily a bunch of Python scripts that run sort of everything for us. Specifically, our molecular dynamics, those are all ran with Python, as well as sort of our electric field analysis. And then my in silico screening is sort of the latter next upcoming steps of my project. So I haven't really gotten nitty gritty in there yet.
Okay, we do have one additional question. Casey, would you like to ask?
Yeah, great presentation, Becca. I just you might have said this and I just missed it, but is there a particular reason why you'd want to target one conformation over the other?
Yeah, so like I said, these are really like newly identified. They do know that form 1, so the 7CV3 form, is like the primary form, so that forms 75% of the time in vivo, whereas 7CV4 forms approximately only 25% of the time. However, there's sort of ongoing studies as to what controls which form is actually present. Sort of a lot of other genes that have sort of two G quadruplexes, there's sort of a difference in sort of like the disease state that requires or that allows them to sort of interconverge. Again, that's just like not known for this right now. So my project is sort of just focusing on how we could target one over the other and then sort of supplementing that further with new biological information as it comes out.
Thank you.
Thank you, Rebecca. We will go on to the next talk, which is given by Emma Cleveland at Virginia Tech. Go ahead whenever you're ready.
Alrighty. Hi, everyone. My name is Emma Cleveland. I'm currently in my junior year as an undergraduate studying systems biology at Virginia Tech. And today I want to take these few minutes to share the research I'm participating in at Brown Lab, which uses molecular dynamics to study the interactions of amyloid beta 42 to better understand the pathology of Alzheimer's disease. To start, I want to discuss what Alzheimer's disease is. It is a complex neurodegenerative disease that affects millions worldwide, and it's characterized by cognitive impairment and memory loss. One of the key players in Alzheimer's disease is amyloid beta, which is a protein that has the potential to aggregate. This protein tends to form long, sticky fibrils that accumulate into plaques within the brain. These plaques are a hallmark of Alzheimer's disease and have been closely associated with the cognitive decline seen in patients. All right, you're good to go into the next slide. We've used a high-performance computing technique called molecular dynamics, which allows us to observe the behavior of amyloid beta 42 and its interactions with lipids. This would have been practically impossible with standard computing resources. With high computing resources, approximately 250 nanoseconds of simulation were performed over 6 days. VA-WHPC significantly accelerated the research, enabling us to run extensive simulations over extended periods of time. So going back to the whole lipid topic, you might be thinking, why are we investigating lipids if the aggregates are composed of amyloid beta 42? This is because the interaction between amyloid beta 42 and lipids is hypothesized to play an important role in Alzheimer's pathology. The lipid chaperone theory suggests that when lipids interact with amyloid beta 42, they encourage the formation of toxic oligomers, which can then be guided and inserted into cell membranes, causing destabilization and ion leakage. Our research specifically uses POPC, a lipid commonly found in cellular environments, as the model-free lipid in these simulations. In our research, we've discovered the interactions between hydrophobic regions of amyloid beta 42 and the hydrophobic regions of POPC specifically form between the residues 16 through 22 and 30 through 35 of amyloid beta 42. We have also observed the POPC acting as a magnet, attracting the amyloid beta 42 peptides, furthering the interactions. The more interactions that occur, the more likely the formation of beta sheets. The increase in beta sheets can lead to greater aggregations and a higher chance of forming toxic oligomers, ultimately contributing to the formation of amyloid plaques in the brain. Altogether, this research aims to collectively analyze the oligomerization events of amyloid in the presence and absence of free lipids, providing atomistic details that can be used to understand the pathology of and prevention of an array of neurodegenerative disorders. All right, that's it. Thank you for your time and attention.
Well, thank you. Are there any questions? Don't be shy. Oh, we have a question. Go ahead, Anshu.
Excellent. Thank you. Thank you for your presentation. Quick question. I apologize for missing this. I'm sure you mentioned, but you mentioned the HPC sped up the computation a lot. What sort of a computation were you trying to do? Were you trying to simulate like how these molecules were bumping into each other, or was it something more at the chemical bond level, or was it a mixture of both, or?
Yeah, so we started the simulations out where we put both peptides and lipids into a box of a system, and we let it's a molecular dynamics lets it run and see the interactions that occur between both the peptides and the lipids on like you said, just on a visual basis, but also like understanding the chemical interactions that are going on there.
Fascinating. Thank you.
All right, Alistair, another question?
Yeah, I'm curious, what software were you using for your molecular dynamics simulation?
GROMAX.
GROMAX. Okay, thank you.
Yes.
Okay. All right, well, thank you. We will move on to the next talk. The next talk is given by Raina Fisher from University of Virginia. Get this up here. All right, very good. Go ahead whenever you're ready.
Awesome. Thank you so much. I'm Ronnie Fisher. I'm a third-year undergrad at UVA, and I'm studying global public health and data science. My research is part of an ongoing project within the UVA Biocomplexity Institute's Network System Science and Advanced Computing Division. And my mentor for this project has been Ben Hurt. For the past 2 years, we've been building a data set on county-level non-pharmaceutical interventions, or NPIs, for COVID-19 by manually pulling data from local sources. As I'm sure you all are very familiar with, during the height of the pandemic, there were all sorts of different mandates that varied on a state, federal, and local level. Having local and specific data is really important for analyzing trends in viral and human behavior, but most data sets of this type are only reported on the state or federal level, which is why our work is significant. So you can go to the next slide. We looked at 6 types of mandates: school closures, college closures, religious closures, mass mandates, business closures, and lockdowns. And we looked at these across 6 states. So that's Minnesota, Arizona, New Jersey, Washington, Hawaii, and our home state of Virginia. And this past summer, I worked on analyzing the data set that our team has spent a lot of time building. So here on the left, we have a chart that shows the number of mandates in place versus the number of COVID cases by subtype of mandate. As you can see, the only category that really kept up in volume with the number of cases throughout the pandemic were mask mandates. This makes sense with what we know. Mask mandates are less restrictive, and mandate fatigue set in pretty quickly across the US. So knowing that mandate activity is not completely dependent on viral behavior, I went to look at some other variables that could correlate to this. We didn't find a lot of strong correlations, but school and college closure length had significant correlations with political affiliation. So counties that tended to lean more Republican tended to have shorter mandates. We saw an opposite trend for lockdowns. The lockdowns tend to be implemented more on a state level than a local level, which could affect some of those results. Using this data, I built a random forest classification tree that took county features and used those to predict how many mandates a county would implement within the first year of a pandemic. The variables I used for this can be found there, but they're location, population density, political affiliation, income, education levels, GDP, and presence of hospitals and colleges. Location actually ended up being the strongest predictor of how many mandates a county would implement, which indicates that there were other geopolitical or cultural factors that I didn't take into account in my model and that I could in the future. The model was accurate about 2/3 of the time, which is okay, but obviously leaves a lot of room for improvement. However, we saw stronger trends for states where we had more data, which is a really positive sign. So right now I'm working on collecting more data to round out our set and hopefully draw stronger conclusions about the relationships between those geopolitical factors, NPIs, and viral behavior. Thank you.
Thank you very much. We have time for some questions. If somebody would like to go ahead.
Quick question. What were you using to analyze this data?
Yeah, so I've been using Python for this. The data set, since we built it ourselves, is not as large as some of the other ones that require high power computing, but yeah, I'm just using Python scripts.
And what statistical measures are you calculating?
There were a lot of different statistical measures. I have a lot more that I couldn't include on the slides just for time's sakes, but a lot of different correlations on individual variables. And then obviously for the model, we had accuracy, precision, recall, all of those values on the screen.
All right, 2 more questions. We have Grace first.
Yeah, sorry, this is not a question related to the content of the talk. I have Rania down for costume contest. Are you actually wearing your costume?
No, I came from class and didn't have time to put it on, unfortunately. I'm sorry, but I did plan to.
That's okay. Thank you.
Okay, then Catherine.
I do have a content related question. Great talk. And I'm actually also with the Biocomplexity Institute, but I'm with the Social and Decision Analytics Division. And I was curious, you said that one of the novel pieces of this work was that it's at the county level rather than the state or larger geographical level. So did you utilize NSAC's synthetic populations at all for this work? Or is this just strictly on the factors you have on the slide?
I haven't myself, but I'm not the only one who's working on analyzing this data set. I'm sure there are other people within biocomplexity who have been.
Gotcha. Just curious. Thanks.
Yeah, thank you.
Okay, well, thank you very much. We will move on to the next talk. The next talk is given by Anna Schmedding from William and Mary, fantastic school. Here we go. Oh, I should have preloaded the. Anyway, here we go. Go ahead whenever you're ready.
So I'm Anna Schmedding. I am a PhD candidate in the computer science department at William and Mary. Today I'll be talking about our work, Transient Faults in Autonomous Vehicle Neural Networks, Resilience Evaluation and Mitigation. Next slide, please. So first we have to talk about what we mean by a fault in an autonomous vehicle framework. Specifically, we're focused on faults in DRAM and memory, where a 0 gets flipped to a 1 or a 1 gets flipped to a 0. These can happen from a variety of different causes, such as cosmic radiation. And these can happen either as single bits that flip or multiple bits that flip. We are more focused on the multi-bit case because these can't be corrected by existing mechanisms in memory and computing systems. And then we model these faults through fault injection. So specifically, we look at the learning by cheating or LBC framework for autonomous driving. And within that framework, we focus on the machine learning model. This is a convolutional neural network where an input image is processed through convolution layers. Then there are 4 branches corresponding to high-level control commands like turn left or turn right. And then 5 waypoints are output for the car to follow. Within this, we inject faults into weights in this model. And because there are so many weights, there are millions of weights in this model, we have to be careful about how we select fault sites when we're looking for these critical fault sites that affect the safety of the vehicle. So in order to address that, we developed Taylor-guided fault injection. This uses the Taylor approximation of each weight's contribution to the accuracy of the network as a proxy for its criticality to the safety of the vehicle. So then we inject these faults in the weights, and then we run simulations with a car in the simulated road, and then we see what happens to see whether or not this causes an accident or some other sort of safety violation. Next slide, please. So first, we evaluate just the resilience to the fault without any sort of mitigation measures to help improve the safety of the car. So that's what we see on the left here in the form of this CDF. So first, as a baseline to compare to our TGFI, we do random fault injection. We randomly select a fault site, we inject a weight, we see what happens to the car. Based on the height of this green line, we can see that this does not find very many critical fault sites, which is to be expected when there are millions of weights. When we look at the top 500 weights as found by TGFI, we find significantly more critical weights, and TGFI top 50 finds the most. Across all of these, the hazard occurs in just about 2 seconds, which means that we really have under 2 seconds in order to mitigate the fault. So when we want to find a fast mitigation measure, we look at Ranger. So Ranger is a method that restricts ranges of values in the activation layers of the network, where basically if something is out of bounds, it gets clipped back into the bounds in order to preserve the accuracy of the network as best as it can. So we do comparisons with and without Ranger for various weather conditions and various road conditions. So when we see the hatched lines, this is without Ranger, we can see that there's a high percentage of hazards. And when we apply Ranger, this improves significantly. However, challenging scenarios such as rainy weather or sunset weather, where it was already difficult for the vehicle to see the environment, that does not improve quite as much. But overall, we do see a great improvement. So in conclusion, we see that the learning by cheating framework is vulnerable to these faults. This TGFI finds these safety critical fault sites effectively, and Ranger mitigates them very well. Thank you.
Thank you. We have time for a quick question.
Yeah, could you just quickly explain what learning by cheating is?
Oh, so that's a framework for autonomous driving. So it's the learning by cheating name comes from the way in which this model is trained. So it's an end-to-end framework for self-driving. Is sort of the short version of that.
I was just wondering if specifically the cheating part, how does that?
So that's based on, so it's trained with a basically a teacher and a student model, and it's cheating is sort of in air quotes here, but that's basically because it's trained by using some input from the environment instead of just looking at what it's seeing. So some input about like the map that it's on, this kind of thing.
Okay.
Okay, one more quick question, Grace Chamberlain.
Yeah, I was just wondering how you implement conditions such as cloudy and rainy into your simulation.
So we use the Carla simulator for autonomous driving, which is based on Unity. And within the simulator, there are already different weather conditions that you can select. So we didn't actually have to implement those ourselves. We just had to implement the framework into Carla and then do our evaluation.
Oh, thank you.
All right, well, thank you very much. Forgive me for setting up the next talk here.
Thank you.
Thank you. So the next talk is given by Sabiha Salma from George Mason University. And go ahead whenever you're ready.
Thank you. Hello, everyone. My name is Sabiha Salma, and I'm a fifth-year computer science PhD student from George Mason University. Imagine you are a real estate contractor who renovates old houses, and you are constantly trying to make the changes and updates that clients want. Now, think of identifying the changes required between features of old homes with those in new design requests, creating updated blueprints, and renovating the new home applying these changes. A lot of effort in a competitive market. That sounds challenging. Mobile app developers face a surprisingly similar scenario. That is, they constantly need to update the graphical user interfaces or GUIs of their apps to meet both changing user demands and design guidelines of new versions of mobile operating systems. Think about how often your phone updates. When developers need to update a GUI, they typically create a mockup or image of the GUI they want to implement, often in photo editing software to help guide their coding. Currently, developers manually identify these changes between existing interface and mockup of upcoming release, create new app metadata or blueprints for mockup interfaces, and implement these updates into the code base akin to renovating a property. To help ease the burden of implementing these changes and to help free mobile app developers to focus on fixing bugs or adding features, my research introduces GUIVO, a computer vision-based automated approach for GUI evaluation. It aims to assist developers in the process of GUI evaluation by detecting changes in GUIs between existing releases and proposed mockups, and automatically generating updated GUI metadata for the new release. GUIVO works by using advanced computer vision technique to automatically understand the structure of user interfaces and implement necessary changes to evolve an app user interface. We have found that GUIVO works quite well. Our evaluation demonstrates that GUIVO can detect GUI changes with over 85% accuracy, and the generated GUI metadata closely matches the expected structure, saving developers days to weeks of effort. You can visualize GUIVO automating for user interfaces what real estate contractors manually do for home renovation. I hope my research will help mobile app engineers automate the tedious parts of software development so they can focus on adding delightful features that you will love using every time you use your phone. Thank you so much.
Thank you. Well, we have time for a question.
Yeah, I'm curious, what were you running this on, this GUIVO?
So we have done everything on Python.
I mean, what hardware, what resources were you using? Were you using a cluster, or?
No, it takes very low resources, so I did everything on my own machine. That's all.
Okay.
Yeah.
Okay, Grace, another question?
Yeah, I was wondering what kind of evaluations were used to evaluate how well the your proposed method works.
So basically, we have done different types of evaluation. For example, Guivo automatically generates the metadata or hierarchy. So for that, we used three-edit distance measure metric to quantify how much difference there are in between the generated metadata and the ground truth. And for the screen matching, we have used precision, recall, F1 score, accuracy, this kind of metrics.
Okay, well, thank you very much. All right, our next talk is Alina and Akiva. I believe I pronounced that somewhat correctly.
Yes, thank you.
I tried. So here we go, and you can start whenever you are ready.
Thank you. At many universities, rigorous research demands extensive calculations. Students and faculty, as it turns out, make wild guesses on the number of resources their job needs, requesting multiple CPUs for a single threaded processes or requesting gigabytes of memory for a simple calculation is what takes up the resources of the cluster and what prevents others from using it. Using HPC is a challenging task, especially if one is unfamiliar with computer science concepts of resource allocation, but it can serve you well if used carefully. How can you execute your job the fastest? How to ensure that your job takes as much CPUs and memory as it actually needs? Well, let me present you with a transformative solution that provides real-time insights into the availability and performance of the computer cluster. All you need to do is to enter the map. Next slide, please. This is a visual representation of a cluster status. It simplifies the complex task of understanding resource availability. Could we switch to the next slide? Enabling users to not only pick the nodes for the fastest job execution, but also to track the node's occupation before and after submission of the calculation in order to learn and make better predictions next time. Here at the University of Richmond, we have 13 nodes, each bearing the name. Yeah, I don't think I see the slide, sorry. Thank you. There. Here at the University of Richmond, we have 13 nodes, each bearing the names SPDR, which have 52 cores and a memory capacity that ranges from around 300 to around 1500 gigabytes. Next to each node's name, the map tells a story. It reveals how many cores out of the formidable 52 Slurm, which is a scheduling agent, has allocated based on user requests. The columns labeled allocated indicate the cores and memory that users initially requested for their tasks. It's not uncommon for users to either underestimate or overestimate the resources their program needs, and this is faithfully reflected in the used columns. The numbers here indicate how many resources are actually being used. The map's visual cues are as informative as they're striking. A sea of green indicates that a node's load is comfortably below 75% for both memory and CPU usage, painting a serene picture of resource availability. A node bathed in yellow, however, sends a warning signal. Its memory or CPUs are under significant load, with usage surpassing the 75% mark. And then there is red, the color of anomaly. It shows that the node is either offline or users' calculations as a result of underestimation require more resources than requested. The allocated numbers may seem reasonable, but the used values tell a different story, a story of a cluster oversubscription. In essence, this program is more than just a helpful tool. It's a dedication to enable academic communities to reach new heights in research via timely job execution. My name is Alina. I graduated from the University of Richmond with Bachelor of Arts in Computer Science degree in May 2024, and I'm looking for job opportunities. Thank you.
Thank you. We have time for a question or two.
Yeah, I'd like to ask, how are these statistics gathered on the job? Are these real time or these
Yeah, they're real time, and the page can be updated by pressing one of the keys on the keyboard. And the script, it's written in Python, gets executed once upon the click of the button. So you can get availability of the cluster in real time. And so what the script does is it goes into each node and executes the command that gathers all the necessary information, and then my program puts it in order basically and puts it in the table. And also, accessing of node happens simultaneously.
Are these nodes shared, or are these unique? Are these nodes basically when you get a job running on your cluster, does it get a node to itself, or does it share jobs on the node?
Some of the jobs, some of the nodes are community nodes, and everybody can use them, everybody who has access to cluster. And other nodes, they belong to certain faculty members, and so their students can only use those.
Okay, but I mean, I say you've got a shared node. If someone's using it, can anybody else use it at the same time, or is it dedicated to that person until they're finished using it, and then another person gets to use it?
Yes, as long as there are resources available, so people can use it.
Okay, so this information you've got, it's really job level.
Yes.
It's based on node statistics. Is that right?
Yes, it's
For
based
the whole
on
node. Okay. Thank you.
Okay, well, thank you very much. We will move on to the next speaker, which is Shawal Khalid from Virginia Tech. Let me get that up here. Okay. Whenever you're ready, please go ahead.
Hey, Eric. So I updated my slides to the 2 minimum.
Okay.
So like, yeah.
Okay, very good.
Do you want me to like, cause it's shared with you, do you want to like download that real quick, or do you want me to like?
Oh, did you give it to me?
Yeah. So like I shared it with you.
Last night?
Yeah, last night.
Yeah, that's the one I have right here. The 5 slides. I didn't see a new one from you.
So I updated this one. So it's on Google Slides. So I guess you need to download.
Okay.
Yeah,
Sorry.
I
I'll have to
will.
find it.
If you have them ready, can you just share your screen?
Yeah, why
Siobhan?
don't you just do that?
Yeah, you want me to share my screen?
Yeah, let's do that. Let me stop sharing.
Okay.
Okay. Whenever you're ready, go ahead.
Okay, hi everyone. I'm Shawal Khalid. I'm a PhD candidate at Virginia Tech. I'm studying computer science. Our lab has a very cute name, Code World, no blanket. I'm currently working on blockchain-oriented software engineering. So the main motivation behind working on these blockchain tools is the emergence in blockchain technology, and it's like expanding. It's not just cryptocurrencies anymore. So it's emerging in different fields, and there are a bunch of different blockchain development tools that are coming in action. So we wanted to see if like the traditional software engineering methods like Agile, Waterfall, Scrum, and other software engineering methods go well with blockchain development, or we kind of like need different set of methods for blockchain development. So since these traditional methods have been tested for years and years for regular software development, and they are proven to be very good, specifically Agile methods, software developers perform very well using these methods, and they kind of like talk about the benefits of using these software engineering methods, of like keeping them on track and just like going going towards their goal in a better way in while using these traditional set of methods. So our main goal was the question was, can blockchain leverage these traditional software engineering methods effectively, or we just kind of need a new set of methods, or can blockchain developers just work with the same software development methods? So for that, we just completed working on a project that is software engineering approaches adopted by blockchain developers, and it was also recently accepted into the software defined systems conference. I presented there last week in Texas. So for that, we just surveyed a bunch of blockchain developers and we asked them whatever projects you guys have worked on. So did you have used any of these traditional software engineering methods? So from the outcome, Agile seemed to be like the top choice among the blockchain developers. So it kind of like is similar in the traditional software development, like Agile is the top choice among developers. And then in blockchain, kind of
Eric, is this the speakers?
Yeah. I think your audio is really getting bad.
It's similar.
So your audio, we can barely. Oh man.
We lost her?
Yeah.
Okay. What we can do is we can come back to the end.
Yeah. Well, there's one more, so
Yeah.
maybe we can do that, and if she can come back, that's great. Okay, so let me.
Shawal, we lost you for a moment, so we lost your presentation.
Yeah. I'm so sorry. I think my internet, it just went buffering and it got reconnecting.
That's okay. What we're gonna do, we're gonna move to the next speaker, and then we're gonna come back, circle back.
Okay.
Okay.
All right, let me pull up the final speaker will be Grace Chamberlain from James Madison University. And I'm very curious about this one. Okay. Whenever you're ready.
Eric, are you sharing your screen?
Oh, yeah, that's important, right?
Yeah.
I forgot I had stopped. All right.
No worries.
Okay. Should be good now.
All right. Good afternoon. Good afternoon, everyone. My name is Grace Chamberlain, and I am currently a sophomore physics student at James Madison University. 14 years ago, I dressed as a battery for Halloween, and today I am going to discuss my investigation of materials called high entropy oxides that could improve lithium-ion batteries in renewable energy technology. High entropy oxides, or HEOs, can form lattice structures at very high temperatures. However, these high formation temperatures can make synthesizing HEOs in a lab very time-consuming and expensive. This is where our lab's research comes in. Using HPC resources at JMU and UVA, we can run a software called JDFTX that allows us to model theoretical HEOs and predict their formation temperatures. However, we have to make some guesses when it comes to the HEO's structural characteristics. So how do we know that these guesses won't dramatically impact our predictions? To answer this question, next slide, please. We altered 4 different structural characteristics: the lattice constant, lattice type, symmetry, and size. We then analyzed how changing each characteristic affected the likelihood that an HEO structure would form at a given temperature. So with these curves here, as the temperature increases, the likelihood of that curve goes up, so the likelihood also increases. We see an overlap between curves in all cases, except when the lattice size differs. And this overlap suggests that when we do not have experimental data to reference for an HEO's lattice constant, lattice type, or symmetry, we can still use HPC to investigate theoretical HEOs and produce significant results. When we change the lattice size of our HEOs, as shown by the graph on the right, we find that the curve for the larger lattice falls almost entirely above that curve for the smaller lattice. Therefore, we should use larger lattices in our future calculations because they are more likely to form than smaller lattices. Many of our larger lattices have around 64 atoms, and many of those atoms have over a dozen electrons, which are all interacting with one another. So HPC is necessary for these future calculations with larger lattices in order to account for all of those interactions between the atoms and their electrons. Thank you all for your time and your attention.
Okay. Well, we definitely have time for some questions if we want to go ahead. Let's see here. Oh, I got my screen all screwed up because I'm stopping sharing. Okay, Mark, why don't you start?
So I was going to ask you, Grace, it's curious that you went as a battery. Most kids would not go as a battery. And then it's also interesting that you continued research on battery. What kind of got you so fascinated in batteries?
So honestly, when I was a kid, I have no idea what made me want to decide to be a battery. I just told my mom one day that I wanted to be an Energizer battery, and we just went with it. Yeah, I honestly, I applied for this research project last semester, and I got matched with my professor, and I liked the idea that there were a lot of opportunities to learn how to use high performance computing, because I didn't really even know what a supercomputer was or how to code or anything, and I wanted to learn more about that. And so the batteries just kind of came along with it, but it was kind of a cool full circle moment, I guess.
All right. I have a question. The JDFTX code that you use, what sort of like interactions? Is it density functional theory, or is it something harder than that, easier than that? How do you do the atomic interactions? Like what level of theory?
Yeah, it's a density functional theory software. So it accounts for the quantum mechanical effects. Yeah, JDFTX, I believe, stands for Joint Density Functional Theory. Yeah.
Okay. Are there any other questions? Okay, well, why don't we let Shawal Khalid put her slide back up. I will stop sharing. And if you would like to do the second half of what you were talking about, I think we'd all appreciate seeing it.
Yeah, sorry about that. You can go ahead and share my screen. So are you guys able to see it now?
I can see it.
Yeah, so I'm not sure when I cut off, but that's what I'm working on right now. So like just working on these traditional software engineering methods and seeing how these fit into blockchain development and how blockchain developers want to see, or if they want to see the need of new software development methods or different tools that can help them with blockchain development. There are also like some other work that I worked on, which was kind of like revolved around crypto. So understanding the GitHub developers and their development activity across GitHub in comparison to Twitter influencers. So the goal was we kind of like collected a data set of 100 popular tweets from Twitter from like these top influencers like Elon Musk and some others that keep talking about cryptocurrencies and stuff. So we kind of collected a data set, and we just wanted to see when these famous influencers like Elon Musk post a tweet about Dogecoin or any other coin, does it have any impact on the development activity of software developers across GitHub? So we just went into their GitHub repo and see if there are any forks or any changes are made to the repositories. So that was also one of the projects that we worked on. And besides that, if you guys have any feedback on like this topic, or if you guys have any experience working with blockchain development, and you guys can give me any feedback, I would really appreciate it, because I'm still like figuring out my current research topic, because I just finished these 2 projects. So I'm like working on starting the next project. So I would appreciate any feedback or any questions. On the next slide, I just have my LinkedIn QR code if you guys want to get connected. So yeah, that's all. And sorry about the cutoff.
Oh, no problem. All right, we have time for a question before we wrap up. I was wondering if you mentioned right at the end there. I know very little about this topic, and I was just curious. You had mentioned blockchain and the use of like changes in a GitHub repo. Could you give like maybe another example where like this is like effective, where this sort of thing can help you with you know, the problems that we're looking at? Or yeah, just like what's the typical like example people give?
So you mean for the last project that I mentioned, the crypto one?
Well, I just meant in general, like where can blockchain, like what do you typically, how do you employ blockchain in these problems? Oops.
So like the main goal, you know, like the main motivation, I would say, is since like blockchain have this kind of concept of decentralization and stuff, and since we all like face issues with like centralized systems, and like there were like some other things that happened back then when like GitHub is not centralized, right? Is not decentralized. So like all of the data was taken down before. So the main motivation I would say behind working with blockchain development tools is its decentralized nature, because it's kind of like reshaping industries, and there is like a huge investment that was made in blockchain, like around 3 billion. And there was an increase in its development tools, IDs, and frameworks, which kind of like reflects its growing nature. So there is kind of a need of understanding its impact in the software engineering community, because it's getting popular because of its decentralized nature. And it's just getting more popular, and it's not just cryptocurrencies, like people are building systems on it and stuff. So that's why, I guess, I'm not sure if that answers your question.
No.
But yeah, that
It does. Thank you very much. I think we need to wrap up. We're about 5 or 6 minutes over. I just wanted to thank everyone, and we'll give everybody a round of applause for all the nice talks we had today. Okay, I'm the only one who'll clap. All right. And just thank you very much. I think Grace posted, or no, hey,
Both me and Haley posted something for everybody.
Okay.
Please kindly vote for the best talk at the link that I posted, and Haley would like some feedback on the Google Forms link that she posted. I'd also like to announce that originally there were 3 best costume contestants. 2 did not manage to come with a costume. So the winner of the costume contest is Eleni Adam.
Awesome.
Yeah, I was quick. Thank you.
Okay, everyone.
So the poll for the best presentation is still going to be open for the next couple minutes. Please put your vote in.
Okay.
You put your vote in so that we can announce here who the winner is. So please, we really appreciate your effort. And you just need to enter the name, I guess.
It looks clickable.
Yes.
Okay.
Let me make sure the poll has not closed yet.
Says waiting for your presentation to begin.
Yes. So I think that you have to write your name as the person that is voting, and then the rest is going to be just choosing.
So what if people refresh their screen? Does it still?
Yeah.
Huh. Okay. So it might have stopped the poll might have stopped itself. So let me reactivate it and give people another minute.
Okay.
Okay, so it should be reactivated and people can. If you have not already voted, please vote. If you have already voted, please refrain.
And yeah, oh, the names are right there. Okay, awesome. Yay. So
Okay.
it's allowing only one vote, right?
Yes, everybody can only vote once, yes.
Okay. And only for one person?
Only for one person, yes.
Okay, perfect. Thank you, Grace.
Of course.
And everyone, we have a feedback form here that Haley just posted. We could just gonna repost it again. And we really would like your feedback. If you could take a few minutes, you know, a minute or two just to fill that. This is something that we want to continue doing, lightning talks. We're trying to find better ways to do this and trying to figure it out. If there is an opportunity for an in-person event, maybe half day where we can all be in person and have some lightning student presentations too going on, something that we're thinking for the spring. So if you please have any feedback, please add it there to the form. If you don't feel comfortable with that and you want to send an email to one of us, feel free to do that too. And we will try to do our best to accommodate the best so we can for the next coming events. Grace, let us know if we have already.
Yes, so a winner and a runner-up have been chosen. And I'm pasting the screen image into the Zoom chat. So I'm happy to announce that our winner today for best presentation is Rebecca Fogarty from Virginia Tech. And runner-up is Grace Chamberlain from JMU. Congratulations.
Congratulations. Great job. Thank you, everyone. Thank you.
And I'll be in touch with all 3 winners about their prizes. And I'll be in touch with them.
Thank you, everyone. And we are really past the hour, but we really thank you for being here. We are planning and concluding the events for the year with this lightning talk. If there is any one of you that is actually attending the supercomputing meeting, please get in touch with us and we can provide you with some information. And thank you all for your time. Thank you for being here. And we hope that you remain connected with the next events for next year. Thank you, everyone. Thank you for being here. We appreciate your time.